Hello and welcome to Biology Interest. Today we're going to be looking at mitosis. This is essentially the way a cell would divide normally when it's inside your body and it's not becoming a sex cell. And it essentially just splits into two, and you have two daughter cells which have exactly the same DNA, unless there's a mutation, as the parent cell. So we're going to draw a diagram to represent what actually goes on in the cell when it's going through mitosis to hopefully make it clear. So the first step is interphase, and that's essentially when the cell is going about its normal daily business, producing proteins. You can't see the chromosomes, but the DNA number is replicating. It is doubling its DNA while the cell goes about its normal business. And so you could basically represent that as, let's draw it on my diagram. And that's essentially just the cell, bit DNA. You can't see anything in the nuclear envelope. That's what this is, and that's the nucleus. You can't really see anything. Nothing much is going on in terms of DNA replication. So after interphase, we go on to prophase, which is the first step in mitosis proper. I like to remember prophase, pro like before almost. So that's quite a good way of remembering it as the first step. So if we draw the cell again, this time we actually have some chromosomes visible inside our nuclear envelope now. So we got two chromosomes here. So they're visible. And so that's prophase. Basically, the chromosomes begin to become visible, they condense almost, and the nuclear envelope starts to break down. That's this thing around the chromosomes here. And so that means we can now go on to the next step, which is metaphase. So if we draw our cell again, this time the nuclear envelope is completely broken down, and the chromosomes have started to line up along the equator, almost. And then the spindle fibers have formed, which are going to pull the chromosomes towards the pole in the next phase. So they're formed, and they're attached to the centromeres on the chromatids, which is the center of the chromatids. So that's metaphase. So once we've got the spindle fibers attached to the center of the centromeres at the center of the chromatids, it's going to begin to pull them apart. So each chromosome, half a chromosome called a chromatid, is going to get pulled towards each pole, almost. So that's going to happen here in a process called Anaphase. So each chromatid, which is half the chromosome, is going to get pulled towards each pole by the spindle fibers. And this ensures that you have the same DNA being pulled towards each side, the same genes almost. So once that's happened, we then go to telophase, which is when the cell almost splits in half as the new nuclear envelopes form, almost. So the chromosomes have been pulled apart to each pole, and when they reach each pole, they condense. They become unwound again. They uncondense, and new nuclear envelopes form, like shown here. And then the cell kind of splits in half, and becomes two separate daughter cells. And then they begin to go about the business of interphase again, doubling their DNA and producing proteins like they normally would. So that's mitosis, the process of mitosis. I hope that's been understood clearly. Stay tuned for more videos. Please like and subscribe. Thank you.